who've joined in this session. Welcome to the International Pediatric Association session, and it's my great pleasure to welcome you to this highly anticipated session on the biology of nurture, where genes meet environment. As pediatricians and child health professionals, we have long understood that a child's development is shaped by far more than genetics alone. Today, we'll be listening to the expert teachers on the interplay between biology and environment, and really thinking about how that shapes the trajectory of health, growth, and resilience across their lifespan. This session brings together a distinguished panel of experts whose groundbreaking work really bridges neuroscience, genomics, and child development. And so we warmly welcome you to the session. So just going through the rules and regulations. If you have questions for the speaker, kindly put them in the Zoom chat feature. And to obtain the certificate of attendance, participants are required to attend the entire duration of the webinar and fill in a feedback form, which will be distributed at the end of the event. Please note that the Zoom closed captions are generated by AI, and there may be instances where the translations are incorrect or misleading. We appreciate your understanding with this matter. So today's session, I'm proud to introduce the moderator, Dr. Takwanda Chimwawa. Dr. Takwanda Chimwawa is a consultant neurodevelopmental pediatrician at Zomba Central Hospital in Malawi, and leading a multidisciplinary team in neurodevelopmental care. His interests include cerebral palsy, epilepsy, autism, and early childhood development, and he also teaches postgraduate pediatrics at Kamuzu University of Health Sciences. Welcome, Dr. Chimwawa.
Thank you, Susan, and thank you, IPA, for giving me this opportunity to moderate this very important session. So we have a series of three distinguished guests, we're going to talk these topics. So I think the first one is Dr. Catherine, I think, is the first speaker. Okay, so I think the first speaker is Prof. Haddad. So next slide, please. So Prof. Haddad is a professor of neonatology and the president of IPA. He's a distinguished pediatric leader dedicated to improving neonatal and child health globally through advocacy, education, and international collaboration. So welcome, Prof. And I think the floor is yours. You have 15 minutes for your talk. Thanks.
Thank you. Thank you very much. I'm so proud and honored to be at this distinguished panel of moderators, speakers, and also attendees. Thank you, those who are joining us. So, Madmuta, could you, so could you put it in slide? So I will briefly talk you on this is my point of view on epigenetics and impact on human development. Next slide. So is it nurture or nature? And there is a big importance through this what I call this cartoon. From genotype to phenotype, your life course is headed by a number of factors: epigenetics, nutrition, culture, and lifestyle. Next slide. Next. And DNA is not your destiny, as you may see in the next slide. Next slide. You see, we have three important persons in the world: Miss World, Mr. Trump, and the Korean guy. They have the same DNA, but their phenotype differs. That's very important to know. Next slide. So what determines the phenotype? It's genetics, it's epigenetics, and its environment. Next slide. So in my point of view, there is an active interaction between the epigenetic, between health, between environment, between nutrition and behavior and cognition. And that's what I call the epigenetic wave that goes through your life course. Next. So I will briefly overview the epigenetic process because there is more specialized speaker on that. Epigenetic process is due to either modification of the histone, DNA methylation, or the third one I missed it. Never mind. Next slide. So you are, every child, every human being is inherit his genes from his parents, but experience during life would rearrange certainly his phenotype. So there is a basic background, what you get from your parents and what experience and environment will shape your genotype, your phenotype. Next. So there is certainly positive experience that support your relation and will enhance and boost your development. But unfortunately, on the other side, there is a lot of negative influences such as environment, toxic, and so on. Next slide. So those are the multiple epigenetic factors that can influence your phenotype. The diet, the exposure, and there is a lot, microbiome, alternative medicine, and so on and so on. Next slide. So this is some of what we do know actually from the stress. If you are subject to one stress, there is clear evidence that there is a process of epigenetics and there is a methylation process that is ongoing. However, we are resilient for the first acute stress, but unfortunately, if those stresses continue over life, unfortunately, this will lead to stress vulnerability and psychopathology. So we have actually proved that stress will induce epigenetic factor, and if it continues over life, unfortunately, you will end up with a mental health problem. Next slide. Next. We have proved that poverty also has an effect on the developing brain. Look at the difference between the neurons of a normal infant and those who are subjected to poverty, toxic stress. The neurons are damaged. Next slide. And this is next slide. And this is well seen here in this cartoon where the circulation of this is a PET scan where circulation in the hippocampus are lacking in the exposed child, whereas in the normal child there is a normal circulation and blood flow. So clearly poverty, stress, and so affect the development of the brain. Next slide. And this is also well known that significant adverse impact during childhood will lead to, unfortunately, developmental delays, and also to adverse experience will lead to non-communicable disease later in life. Next slide. We have also seen that in those two papers that racist neuronal connectivity and epigenetics are affected among Black women if they are subjected to continuously racism and discrimination. And here is another study showing that between the Black and white there is difference in this is the Rodan study, and these are the white and the Black people who are subjected, unfortunately, to racism have a lot of non-communicable disease, more than the white man. Next slide. And what we do know from epigenetic and pollution during the development, indeed, pollution will lead to demethylation, as you may see here, and will certainly affect all your development. Next. Next. This is the ER visit increase with ozone concentration, as you may see in this meta-analysis. This is an effect clearly an effect of environment. Next, toxic and pollution. Next, this is also what we do know actually from the genetic variant and the environmental risk factors that lead to autoimmune disease, inflammatory disorders, and cancers. Next, and we do know that exposure to environmental pollution will certainly affect our cognitive development. Next, what do we know actually from pregnancy and epigenetic? Indeed, those are the epigenetic processes that can be unfortunately under pregnancy. You have DNA methylation, histone modification, and non-coding mRNA, and those are the factors that may induce epigenetic processes. High altitude pollution, low nutrient availability, those are the maternal factors, those are the placental factors, and those are the fetal factors. So we do know actually that epigenetics starts from the womb. Next, and this is what we are actually know, that epigenetic will unfortunately will lead to fetal growth restriction, obesity later in life, non-communicable disease, and so altered neurons and so. Next, we do know that gestational diabetes, maternal obesity leads to adult obesity, and we know that toxins and tobacco and alcohol lead to teratogenesis. Next, we do also know that tobacco and epigenetics, that tobacco influences the epigenetic process even and will lead to translation of epigenetic processes or genes that had been modified through the first pregnancy, the second generation, and the third generation. Yes, a woman who smokes while pregnant and uses epigenetic changes even in 3 generations. Next, C-section. Indeed, actually, we do know that C-section has short-term morbidity, but we do know that they lead to asthma, type 1 diabetes, celiac atopy, and obesity because of the perturbation of intestinal flora, and we do know that the intestinal flora is a major epigenetic factor. Next. Then now, how we can avoid epigenetic waves during our life course? This is the first 1,000 days. We have to avoid every epigenetic factor during the human brain development, and also we have to avoid epigenetic factors that lead to metabolic syndrome. And mainly during the first window of life, which is the first window of fertility, what we call the 1,000 days. If you lack avoiding epigenetic factors here, it's very difficult to catch up in the second window of opportunity. Next, how to enhance the nervous system. There is 2 factors, environment and nutrition. Next, role of environment. Next, indeed, during the 2 years of life, you see how the neurons progresses, and at 2 years of life, you have the same neurons as the adult. So during that period, it's very important to have a good stimulation and to have the power of senses very well used. Next. Power of senses mean during the first NIDCAP and so. Next. We do know that breastfeeding, NIDCAP, and kangaroo methods have an effect on the sensorial stimulation and reduce mortality, morbidity, and have a better behavior and cognition. Next slide. Next, breast milk. It is a very important milk. Indeed, it has 2 major factors: stimulation of power senses, oxytocin for the love bond, and the nutrients, neurological nutrients, immunonutrients, bone and growth nutrients. Next, we do know that breast milk exposure is associated with better cortical maturation in preterm infant. Next, and we do know actually, and this is very interesting, that the breastfeeding, breast milk has potential epigenetic effect on infants' neurodevelopment. This is well actually attested by over 50 articles. Next. Next. So what are the other important nutrients that the brain fuel with? Next. Next, protein is important because it is the main substance. It's the main protein are in the cortex. And you see, whenever you are subjected to malnutrition, your neurons are distorted. Next. Next. Iron. Next. Next, iron is very important for dendrogenesis and all the process of a human brain development. Next. 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 Next, and you see here the difference between two brain, a brain with a multiple neurons, which is a brain who is iron sufficient, and this is a brain with the deficient iron when you have scarcity neurons. Next, omega-3. Next, I will pass on that. Next, next. And unfortunately, we are faced actually with two major problems. Undernutrition and overnutrition. How to balance that? This is the main issue that a neonatologist or a pediatrician is faced during the first years of life. Next. 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 So from genotype to phenotype, your life course is under multiple factors: epigenetics, nutrition, culture, and lifestyle. Next. Thank you very much.
Okay. Thank you very much, Prof. I think with the interest of time, we'll go straight to the next presenter. Can I have the slides, please? So having run through that interesting epigenetics, let me introduce our next presenter, Dr. Catherine Mutinda. Next slide, please. So Dr. Mutinda is a pediatrician and a medical geneticist with Nairobi City County and an adjunct lecturer at the University of Nairobi and a member of KEMR CELU committee. She has a lead key child health initiatives, published widely, and advocates for ethical genetic practice in Kenya. Welcome, Dr. Mutinda. The floor is yours. Thank you very much.
Thank you very much for the invite, and I hope to not confuse you more, but just bring the genetic angle and an overview of epigenetics and how this whole topic becomes very interesting when it interacts with our health. So my outline is very simple. I'm going to go through definition of some terms so that we are all talking in the same room, and then look at the epigenetic mechanisms and answer my president's third modality, which slipped his mind, but we're going to look at it as well, and slide in effects on health that I expect most pediatricians have come across that are explained by epigenetics. So the idea of epigenetics started around the mid last century. And then we were thinking that there's a branch of biology that has a causal relationship between the gene and their products. And therefore we see a phenotype. But there's something wrong with that definition, because we are seeing that the genes have a product already. Then the product is the one that is interacting with the gene to make a phenotype. So a lot of work went into it, and now we accept that epigenetics, just like the word says, epi from Greek, above, the genetics, that's above the DNA, means that there are changes on the gene function, the function of the gene, and these genes are in mitosis or meiosis, and these functions are actually being influenced and they're inheritable. And this influence mechanisms or groups of chemicals that influence how genes are going to replicate or translate are either methyl groups, and they attach themselves onto the DNA itself, or attach themselves onto the histone molecules, a molecule within which the DNA wraps itself around the DNA molecule. So that definition has stood the strength of time and explains a lot about our diseases. So as I go ahead, I'm going to remind my chair that the last thing that he forgot was the chromosomal imprinting or non-coding RNAs as a epigenetic modification. But essentially there are three. There's the covalent modification of DNA bases, methylation being the most studied, of cytosine. Then there's the histone modification with the translational modification of amino acids in the N-terminus of the histones. And they can be either acetylated, methylated, phosphorylated, or buccinated. And then we are going to talk about chromosomal imprinting. Either the autosomes are imprinted or the X or the sex-linked chromosomal imprinting. And that is a form of epigenetic modification around the non-coding RNAs. So let's start with the covalent modification of the DNA. So the most studied covalent modification is the methylation of cytosine in the promoter region of all our DNA sequences. Cytosine, the DNA sequence in an area called the promoter region, we have islands that are CG island, that's cytosine and guanine islands. And the cytosine in this area are sometimes methylated. And when they are methylated, and I've shown the cytosine molecule right there, they become 5-methyl cytosine. And this 5-methyl cytosine attracts regulatory proteins. They can be deaminated into thiamine, which we see in the RNA. But there is an enzyme that is very crucial in the formation of this methylation, and that's the DNA methyltransferase. We have 3 types of DNA methyltransferases, the methyltransferase 3A, 3B, and 1. 3A and 3B are very important methyltransferases that are important in the creation of new de novo methylation profiles within our DNA. But once these are made, to make it firm or to be able to inherit that methylation profile, you need the DNMT1. That one maintains the methylation profile. So as Prof was talking about the different toxins, once the DNMT3A and DNMT3B do a de novo methylation, this can be lost during replication. But once DNMT1 gets involved, that becomes a permanent state and it can be inherited in the next generation. So what are the clinical implications of this that we see from a genetic point of view? As I said, the CG islands are promoter regions, and a promoter region is the one that gives a signal for replication to start. So once you have the CG islands methylated, we attract regulatory proteins, and these regulatory proteins methylate the DNA and therefore make it inaccessible for replication and translation. So basically, when that happens, like I've drawn a DNA sequence here, we have the DNA sequence in the first, second area, and the third area. And these are different promoter regions with CG islands. Once we have methylated them, and because we have DNMT1 involved, this replication and methylation profile is going to be held through and through all the way to the daughter chromosomes and the daughter progeny of this particular person. So what happens if I in fact interfere with DNMT1 gene that maintains methylation? It means that I will not be able to maintain methylation profiles into the next generation. And we see this in diseases that are autosomal dominant, cerebral ataxia, and deafness. But we can also disrupt the enzyme that does the de novo kind of methylation profiles. And when we disturb one of them, specifically the DNMT3B gene, we can end up with immunodeficiencies. I want us to remember these things because they are going to have implications. And I think from what Prof has talked and what our next speaker is going to talk, you're going to see these influences around early childhood development. The next epigenetic modification is the histone modification. Histone modification, the nucleosome is how the DNA molecule wraps itself around a molecule called the histones, which is an octamer, to hold the chromosome in a compact version. When the chromosome is in compact version, of course, it cannot be transcribed, and therefore we cannot see any gene product. So we need the histones to let go or let loose so that we are able to read the DNA molecules for translation and transcription. So when they're in the compact form, they are heterochromatin, and these are the ones that stain when we are looking at them during a karyotype, the gene staining, we see that part of it. And then when they are opened up in the euchromatin area, that's when they are busy replicating and forming products. So when the histone on the end tail terminal of the histone is acetylated, because acetylation is one of the well-defined processes in histone modification, what happens is that the histones let go and therefore allow for transcription of the DNA. As you can see, these two processes are very important. So if you have the histones acetylated, DNA is available for transcription, and therefore you can get products. They are controlled by enzymes, the acetyltransferase enzymes, that deacetylize and acetylize histones in the chromosome, in the DNA structure. So clinically, when we look at it, when the DNA is compacted, it is inactive. When it is let go, or the histone is acetylated, and there is demethylation, then there is replication, and therefore we can see gene products. So one of the genes that is important for acetylation is disturbed. Therefore, the histone is not going to open up. Then we are going to end up with diseases. Specifically, the MEPC gene, we have seen a lot of patients in Kenya with the Rett syndrome. The mutation is on an enzyme, a gene that is very important in the binding of the acetylation of the histone for histone modification, which is an epigenetic modulation of the gene. So finally, I'm going to talk about autosomal imprinting. And this is the mechanism that President, you had forgotten. So autosomal imprinting is a form of methylation that either one respects the parent of origin. So when we respect the parent of origin in our methylation profiles, we call that imprinting. So what exactly happens? So for each adult, female or male, they have regions that are imprinted from the mother, and there are other specific regions that are imprinted from the father. That's the person one. When we go into gametogenesis, these imprints have to all be erased, and then we get into when the imprints are erased. Then from the mother, they both become maternal imprints, and from the father, they both become paternal imprints, so that when we are making our baby here in the embryogenesis, the child then gets one imprinted from the father side and one imprinted from the mother side. This is a very clear explanation as to why we end up with disorders like Angelman syndrome and Prader-Willi syndrome. The paternally inherited chromosome 15 has only the SNRP area on. The UBE3 area, UBE3A area is always turned off for the paternal origin. In the maternal origin, the UBE3 area is turned on. So basically, the mother has specific duties and the father has specific duties. So if you have a microdeletion in the paternal UBA3, UBA area of chromosome 15, the child has no phenotype. There's no problem. The child is perfectly okay. But if that were to happen on the maternal side, then you have a disease called the Prader-Willi syndrome. That said, we also have one more imprinting disorder, and this one is called the X chromosome inactivation. It's a form of non-coding RNAs, and it's a way of those compensation for our X chromosome. As ladies, we have two X chromosomes, so we have to compensate or reduce our X activity This was explained a lot as lionization, but we now know it as X inactivation. And X inactivation, both X chromosomes own an area called the X chromosome inactivation center. And this area possesses an X inactivation specific transcription molecule that is X6, and then the T6 is the opposite. If you read XIST in the opposite, it is actual opposite of the same gene. So both the X chromosome align themselves, then one chromosome randomly produces more XIST gene products. And once it produces this, it allows for that particular strand to have histone methylation to occur all around that strand. The other strand does produce, but not as much. And because it doesn't produce as much, the T6 causes the production of the T6, which is an antisense RNA that prevents methylation of the histones in the active RNA, active chromosome. Therefore, as ladies, we end up having only one active X chromosome. So that's how we inactivate our X chromosome. In the male, we have it, but because we only have one, you can imagine that does not happen as much, but we have some inactivation there. So what happens clinically if we do not inactivate or some of our X chromosomal genes escape inactivation? So we end up with either autoimmune thyroid disease, SLE, and also if we have a nucleoid, that an extra X chromosome that we do not inactivate, like in Klinefelter's, we have an overgrowth syndrome, or in Turner syndrome, where we have only one X and we expected to have another one, and we have hypomethylation, and therefore we have a small girl in the mix. That's quite a mouthful, and I hope I have made epigenetics a bit clearer for our pediatric community. These are part of my references, and thank you very much for the invitation. Over. Thank you, Dr. Takwada.
Thank you very much, Dr. Catherine, for that nice talk, a bit of the biology and a bit of understanding in terms of those. So that brings us to our next speaker, which I think is the last speaker for today. Next slide, please.
So, good morning, everyone. It's Dr. Kanu here. I hope you can all see my screen.
Yeah.
And
so
hear me.
Yeah. Okay. Yeah, you can go ahead, Dr. Khan. Thanks for joining us.
Yeah, no, thank you. So thank you to Dr. Susan for inviting me to give this talk, and for the previous two panelists who have set the stage for the epigenetics in child health. So I'm a pediatrician and also a practicing clinical geneticist, and I trained in New Zealand and Australia, and somehow I found myself in Canada for the last 16 years or so. So I think the previous presenter, Dr. Catherine, really did a great job of explaining the groundwork for genetics. And I realize for many pediatricians, well, you get put off by genetics because most people consider it very complex and theoretical and academic. And so I hope to give you a different perspective and to convey some very key issues with regards to epigenetics and imprinting in childhood disorders. So the learning objectives for this morning are to talk about two groups of disorders, which are the imprinting and chromatographic disorders that cause epigenetic disease in children, to think about methylation and chromatin structure, to be able to produce how they produce these reversible phenotypes, and then to apply the principles to think about how you make diagnosis in these conditions. So broadly speaking, I also talk about the genome or the DNA as something that is static. It's something that cannot change. It's written in stone. the DNA is incredibly long. And so for it to be packed into the body, it needs to be wrapped around structures very tightly. And this packed structure, where it's wrapped around proteins, is what we call chromatin. And the proteins are what we refer to as histones. Now, the epigenome, which is the chromatin and the histones, is a very dynamic situation as opposed to the genome. As Dr. Catherine mentioned, the epigenome needs to be opened up for transcription factors to come and act and express genes, and then closed off when you don't want access to those transcription factors. So unlike the DNA, a lot of dynamic changes are happening in the epigenome. The epigenome also explains why the static DNA can result in such different sets of features in a human. And I take the allegory of the music score written here. Any musicians here in the audience who read that score realize that those notes can be played in so many different ways. And what provides that difference is the scores, the writings like allegando, the crescendo markings, the timing of the musical score. So all these additions on top of that static musical score allow for the same composition to be played so differently by many, many different people. The reason that epigenetics is so important for us to understand is that it can be modulated by our environment, right? So while the DNA sequence tells what cells is what's possible for the cells, it's the epigenome that decides what is real. I'd like to take the example from the Dutch hunger winter, which is a very common example of the epigenome that is used in the literature. And this was a period of starvation during the Second World War in Holland back in 1944 and 45. And what people found when they were following the babies that were born to parents who had been starved is that they had a lot of changes in their epigenome. And in fact, some of these babies grew up to have more prevalence for adult onset disorders like metabolic syndrome that predisposes one to diabetes and fatty livers and things like that. So people realize that early periods in life, so when the fetus is developing, seem to be particularly vulnerable to epigenetic marks that are established. And when reprogrammed, they become stable and dictate the phenotype in the individual for life. So not only is the DNA important, but the environment in which we have grown in, and the environment that we're exposed to as an early child can modulate our health and modulate disease. Now, as Dr. Susan has mentioned, and I won't go through this very in detail, the two mechanisms for switching on and switching off the epigenome are DNA methylation, where these methyl changes are added to particular residues, and modifications to the histone proteins, which can be written by writers, removed by erasers, and then interpreted by readers. There's also higher order chromatin structure, where there are long range enhancers and promoters that coordinate gene programs, complexes that define particular boundaries in the DNA to dictate how the actual DNA is played out. So the whole higher order epigenome is incredibly complex, and it's quite difficult for us to understand. And so it's better that we try and separate it out by using some key clinical examples. Now, folks have known that that babies that have been born following in vitro fertilization are at risk of imprinting disorders. And people have figured out that the environment that the eggs and the sperm and the developing egg are fertilized in can influence the epigenome and result in epigenetic conditions such as have been described by the previous author. So a large-scale Japanese study reported that there was a 4.5-fold increase in particular syndromes that were associated with changes in the epigenome. And this really brings home the fact that it's more than the genome alone that emphasizes how we are physically. It's not only about mutations in the DNA sequence, but it's also mutations in this epigenomic machinery. Now, many of these epigenetic disorders act not by causing a one-time insult, but by continuously exerting an abnormal state throughout life, meaning that this abnormal state persists after that change in the epigenome has been made. And so it leads to the point that perhaps removal of this epigenetic state may actually be able to improve the phenotype in future. So the two major conditions that I'm going to talk about are the imprinting disorders, which have been talked about before. And basically, think about an imprint as a stamp. And so our chromosomes need to be stamped either genes in our chromosomes need to be stamped either male or female. And we need to have the right amount of male and female for us to function normally. On the other hand, disorders of the chromatin result problems with the genes that are writers, erasers, and readers and remodelers. So two distinct different conditions. So with the imprinting disorders, it follows on, as I've said, that you need to have the right amount of maternal and paternal alleles that are expressed, or else you will result result in a disorder. Some of which has been discussed by the previous author. The examples that I use here are Prader-Willi syndrome and Angelman syndrome. Now, both of these conditions are a mirror pair because they result from problems at the very identical locus on the chromosome. However, it really depends on whether you have too much maternal or too little paternal for the disease to ensure, and the conditions are so incredibly different. Angelman syndrome is a condition which is recognized by seizure disorders and an unstable gait. Prader-Willi disorder, on the other hand, is appreciated by neonatal hypertonia, severe failure to thrive in the first year of life, with then changes to this hyperphagia and this inability to stop eating as the child develops. Another mirror syndrome is the Beckwith-Wiedemann and the Russell-Silver syndrome, both again on the same identical chromosome locus LP15. One results in overgrowth, which is Beckwith-Wiedemann, and the other results in growth restriction. So these are some of the features that you appreciate in Beckwith-Wiedemann syndrome. And as a geneticist, these are the features that I look for if a pediatrician calls me to see a child that they are worried about with Beckwith-Wiedemann syndrome. Typically because the child has been born above the normal for birth weight, may experience hypoglycemia in the neonatal period, and has these characteristic features: a big tongue, an umbilical hernia, the classical pits in the ears, the nevus flammis that may be appreciated on the forehead, and also overgrowth. On the other hand, Russell-Silver syndrome is characterized by intrauterine growth restriction, where there's relative sparing of the head growth. And that's why we talk about the fact that the head looks big for the body. Other features include the asymmetric growth, clinodactyly, cafeole macules, which can clinch the diagnosis physically. Moving on now to the other group of conditions with the disorders of chromatin. As you can see, there are these key genes in our body that are important in writing. So adding these marks to the chromatin, removing the marks from the chromatin, trying to figure out what the marks mean, and moving the position. So all these genes here cause very important syndromes that a lot of us in the pediatric world will recognize: Kabuki syndrome, Rett syndrome, which has been mentioned before, and CHARGE syndrome, which is a very common condition. So what is Kabuki syndrome? So typically, Kabuki syndrome is one of these conditions that you consider when a child has some sort of learning disability. The name Kabuki comes from the Kabuki theater actors in Japan who wear makeup with very arched-looking eyebrows, which you can see in this child here, as well. And the other feature that you can notice is the very prominent ears here. So Kabuki syndrome is a disorder of the chromatin where a particular methylation signature can also be detected on the blood on a blood test if you do a methylation array. Now, just remember, Kabuki is not just a mutation in the chromatin gene because it produces a whole range of changes in methylation in the genes, which can be detected by the blood, right? So it's not only a single problem, but it affects such a great number of other genes in the body. And you can imagine this means that it's going to be incredibly difficult to fix or to try and treat one day when we're able to. The other syndrome that is probably more recognized by pediatricians is CHARGE, which stands for coloboma, the heart defects, choanal atresia, the growth delay, genital abnormalities, and the ear abnormalities. For geneticists, when we suspect CHARGE syndrome, one of the very characteristic findings we look for is hypoplasia of the semicircular canals, which is done by neuroimaging. And if you have that, you've probably clinched the diagnosis clinically. So CHARGE syndrome is due to this gene called CHD7, which is a chromatin remodeler. And once again, it affects so many different programs in the body to result in the ensuring phenotype that you appreciate in the individual. Moving on now to how you make a diagnosis of these conditions. So genetics has traditionally been DNA type sequencing, and then with the realization that the DNA is static, and we need to actually look at an active signature. And now we're moving on to RNA signaling, RNA sequencing, because the RNA reflects the active gene expression. And we're also looking at methylation patterns, which are the epigenetic phenomena. And you can now do these methylation arrays or MSMLPA methylation specific arrays to make the diagnosis of many of these methylation disorders. Just remember, exome sequencing will not tell you about any of these disorders because the DNA is static and the change is not in the DNA, it's actually in the epigenome. I want to end my final slide by looking into the therapeutic future of these epigenetic diseases. As I said, the global resetting is not going to be a very feasible approach. And these epigenetic marks are so pervasive. They're cell specific. In other words, you know, certain parts of your brain may be methylated, but not your heart or your liver. And so it's going to be extremely difficult to be able to target the right times of development to actually remedy the situation. There is hope, though. In mice models, folks have shown that the re-expression of the gene MECP2 in the adult mice was able to reverse the neurobehavioral and synaptic phenotypes, suggesting that some of this modulation is not permanently fixed, but maintained by ongoing dysfunction that can be corrected. And so that's given us hope for the future that we may be able to do something to change the lives of these children moving forward. And with that, I'll end my presentation and be very happy to take any questions that folks have. Thank you.
Thank you very much, Dr. Kanu, for that very nice talk. And also thank you all our three speakers for very incredible talks. I think I see only 2 questions in the Q&A. So if folks if you have questions, just type them into the Q&A. So I think the first one, they're saying, what is the role of giving cod liver oil supplement to children as routine? Not sure, maybe Prof, you can try to answer that in terms of.
Yeah, this is a very important question. The DHA, I'm talking about the DHA. There is actually proof that breast milk DHA and even DHA included in some formulas, the meta-analysis have shown that there is a beneficial effect on IQ and on vision in premature infant. But that does not mean that you have the routine I don't know what does it mean routine. Mean routine that you give DHA for every child who is on his development. There is no actually studies really reporting that DHA could be a salvator therapy for example, for autism. It's not the case. We do know also that DHA actually in premature infant has an effect on the bronchopulmonary dysplasia and retinopathy of prematurity. Those are studies that are coming now. And I want to differentiate between the synthetic DHA and the DHA that is normally included or in the fish or in the, for instance, in the food. There is a big difference between those two. So the question is, it's not harmful to give DHA. You can give DHA as much as you can, but you have to also behave that there is a what we call an equilibrium between omega-3 and omega-6, and this also should be taken into consideration. So it's difficult to answer that, but we have some proof that DHA is working. Thank you.
Okay, thank you very much, Prof. I think there's a next one. It's saying, do we understand what condition prompt DNMT1 methylation to occur and hence maintain inheritability? Maybe Dr. Catherine, you want to tackle that one? Thanks.
Okay. What conditions prompt the DNMT1 methylation? So the first thing is we get methylated by the DNMT3A or 3B. And then after that, once that has occurred and it is entrained into our system, and if you look at what Professor Haddad was talking about, severe stress and things like that will change that modification and invoke the DNMT1 gene to make this permanent, therefore inheritable. My colleague from Alberta, anything else more than that? Dr. Kanu?
No, thank you, Dr. Catherine. No, I think there's another question there for you that I might have inadvertently also switched off. Someone has asked about the sex that's affected in Rett syndrome and whether it's a spectrum in autism.
Yes, Rett syndrome affects mostly girls, but you do find some males who are affected. It is when the skewed X inactivation. And what was the next part of that? Yes, some of them do present with autism spectrum of disease.
But in the new DMS5, Rett syndrome is not included in the spectrum autism disorder. In the previous one, yes.
It was.
But
actually,
before, yes.
So this one is now being called what Dr. Kanu is presenting as a chromatinopathy now, classified under neurodevelopmental, under epilepsy. Is it neurodevelopment or something else? Somewhere else they're actually talking about it as a chromatinopathy in classification.
Okay, thank you very much. I think we'll go to the next. Yeah.
Sorry, because I just put up my hand. I know I'm not really supposed to speak, but I think just because we have such exciting speakers, I had a question for Dr. Kanu, and I wanted to ask him as a geneticist. I'm sure in Canada they are like leading in terms of technology. So I wanted to know how AI is impacting your practice of genetics, especially now with AlphaFold and all the work that Google DeepMind is doing. I'm sure there are other organizations doing amazing work. So I just wanted to hear how AI is impacting your work. Thank you.
Yeah, no, thanks for that question, Dr. Rosemary. So there's two parts to that answer. So I run a wet lab as well. I'm a PhD scientist, and we use AlphaFold there. So for folks, AlphaFold is a protein that tells you how proteins, a computer program that tells you how proteins fold. And, if you think about the start of how we got into genetics, we started off with proteins many, many years ago in the 60s. And now we've gone back to proteins because we realize that the DNA doesn't tell us all. right? You can have a change in the DNA, particularly what we call missense mutations, and you don't actually know whether this is a harmful or a benign change. And sometimes by modeling the protein by a computer program, you can notice shifts and how it changes the binding. The way AI is really becoming useful is in the genetic laboratories, where you have all these genetic changes, and the AI then gathers the information from the literature and also runs the pipelines and then produces the report for the human then to go over and read. And so I think that has really cut down the time for analysis. Certainly in the clinic, what we're doing is, as you know, anyone knows geneticists tend to be very pedantic people, and we go through every single detail of the chart. That takes a very long time. And so with our electronic medical systems, AI is also able to comb through the systems and produce all that preliminary workup for us, stuff probably that the resident would do, but now can be done much more quickly. So those are two instances of how AI is helping.
Thank you very much. I think the interest of time.
Dr. Kakoda?
Yes.
Yeah, I just wanted to add to Dr. Kanu that AI is also helping us communicate with each other from different parts of the world and helping us move science a bit faster, especially in research. So we are moving a little faster than we used to move in the last century. Thank you.
Oh, I totally agree with Dr. Catherine. So geneticists in the audience will know that we have a program called Gene Matchmaker. And so what happens when you find a very novel mutation in a gene and you think, I think this is the cause, but no one else has discussed it before, you can upload that particular gene. And if Dr. Catherine has also uploaded a patient, we'll be able to match online. And this used to happen when we went to conferences and perhaps by chance started talking at dinner and said, oh, we have a similar patient. But this Gene Matchmaker has really sped things up and connected us as a community.
All right. Thank you. Thank you very much. I think we can go on and on. I think this is a very interesting topic, but I think for the sake of time, I think I would like just summarize. I think today we have gone through a very, I'll call it the science of early childhood development, where we went from the genes and then to the to what happens when we're seeing the child in clinic. So thank you very much to our three wonderful speakers for the incredible talks. I think all of us have learned a lot. I think now I'll pass it on to the IPA. Thank you very much.
There is two questions that I wish to answer rapidly on the epigenetic
Okay, thanks.
relation.
Go ahead, Prof.
Yeah. The relationship between screen time and autism. I have noticed in my practice that when the children have a spectrum, starting a spectrum of autism disease, if you withdraw from him the screen time, that is certainly an improvement. So screen time, and now AAP and IPA has launched a lot of statements on that. About the care, nurturing care. Yes, nurturing care and behavioral therapies can induce changes in some of our genetically affected children. That's clear because the plasticity of the CNS is reliable and we can count on. Thank you. And sorry for being, it took me one.
Thank you, Prof. Thanks.
Still on time.
IPA. Thank you. Yeah, IPA can take this.
Yeah, so thank you everyone for attending today's webinar. So once the webinar ends, there will be a pop-up on your screen where you will get the link to the feedback form for the webinar. And after that, you can just fill up the feedback form to receive the certificates. Thank you.
Thanks, everyone. Have a good evening.
Yeah. Thank you very much. Thank you, Dr. Kanu, for
Good
honoring
evening.
the invite. Yeah. And thank you, everyone. Bye.
Thanks.
Thank you and good evening.